So today we are going to start with the translation and genetic code. Okay. Now, yes, let's begin with it. Now here you can see in the last session we already studied about how DNA transcribed into RNA, that is simply called mRNA, messenger RNA, right? So now today we will focus how this RNA translate and form of proteins. So we will see what are the actual process of translations, what are the basic requirement for translations, like there are ribosomes and all, how, what are the basic requirement to complete that process translations, right? But before that we need to understand one of the important concept, and that is called the genetic codes. That is genetic code. Now what is genetic code? This is little something which we need to understand. Now you all know that DNA. Yes or no? Yes, you all know about the DNA? Yes. What is DNA? It's a deoxyribonucleic acid, right? Yes. So we all know about DNA. Okay. Now DNA is deoxyribonucleic acid. Okay, this is A. So deoxyribonucleic acid. Now this deoxyribonucleic acid, can I say this deoxyribonucleic acid is often referred as a blueprint of life? Yes or no? DNA is the blueprint of life? Yes or no? Okay. So DNA is called the blueprint of life. Now this blueprint of life, because it provide instructions for what? For building the hair on the top of our head and the toes of our feet. That means of it provide instructions for building all our body structures, right? And DNA is inside the nucleus of every living cell. Every living cell having their own DNA, right? In each and every, that means of in your body there are crore and crores of cell in your body, and each cell having a DNA, right? So it's a double stranded. DNA simply is what? It is a double stranded. Let me draw the picture. So DNA is what? DNA is a double stranded. That means of I can draw one simple figure. Imagine this is, okay, and here, okay, this is just a hair like this, okay. So here, and this small section of the body parts, it contain what? That is skin cell, right? This is skin cell. So in the skin cell, in this skin cell, there is a central nucleus. At the central portion, nucleus is there. Inside there is DNA. Yes or no? So inside DNA, right? And the small section of this, when you observed, you will get the structure of typical DNA, like, isn't it? like this. So what is this? This is the DNA double stranded. So you know that in our body, this particular segment, I can call it as a collagen. So this is a gene. This is for gene for collagen. Okay, this is gene for collagen gene. Okay. Now, if this is a collagen gene, then this gene is going to convert it into mRNA, and then it is going to convert into protein. So protein, that means of which type of protein? It is going to produce the collagen protein. Collagen protein, right? So what exactly I mean, why I draw this picture over here, to understand what is genetic code. Okay, that means of here the DNA has a gene that make up the protein that we need, like the collagen in our skin. Collagen in our skin required skin proteins, and that is called collagens, right? So this gene has a specific sequence of what? Of nucleotide, or we can say the nucleotide bases, nitrogen bases, right? That provide the instruction for building these specific proteins, right? So actually two steps involved in this complete protein translations. The one step that you know very well, that one step is called the transcriptions, and the another step is called the translation, right? So two steps is very important. So let's see what is this two steps. So two steps. The one is transcription, and the another one is translation. Translations. Now here, what is translations? Now you know very well that in our body skin, in our body cell, okay, as I told you that there is a nucleus, and nucleus having lots of DNA structure like this, right? So this is what actually we can say collagen gene is present in that DNA somewhere, like suppose this is a particular segment, so here is a collagen gene present. Just imagine this pink color. Can you be able to see the pink color in that diagram? So this is what? This is called the collagen gene. Collagen gene. And this collagen gene, when it transcript, what will it form? It form a sequence that is called mRNA. So what is this process? This steps is called transcription. Transcription, right? Now after transcriptions, what happened? This mRNA, so this process is what? Transcriptions. So how I can define the transcriptions? The process of copying a segment of DNA into mRNA. So a process of copying the DNA segment into mRNA is what? It is called transcription. And you have already studied the replication process. Yes or no? Yes, in the last session we studied about the replications. What is replication process? You know that DNA is a double helix. So on this double helix DNA, suppose this is the DNA double helix, right? So during the replication, what happened? So you know very well that during this replication process, what happened, that DNA, there is a polymerase, right? Polypeptide chain is there, right? So this chain is going to what? It is going to attach with the simply we can say nucleotide and sequence of mRNA, amino acid in the polypeptide chain, right? So how they are actually converting that mRNA, that we have already studied in the replication process. Yes and no? Just respond to those who are were present. Know the answer. Yes. Yes. So that means of this is what this segment, that replication. So finally this mRNA going to convert into a polypeptide chain. This mRNA converted into polypeptides. Now how this exactly polypeptide look like? So this polypeptide is nothing but it's a kind of, we can say, a different shape of the, okay, so here you can see this is what the polypeptide chain. Hope my this ring circle is visible to you, right? So this is what indicate this is a kind of polypeptide chain like this. Okay, so this polypeptide chain, it is in the form of language or code, and that that language and code, that if there is mRNA, then how this language of code is transcribed. Like suppose there is a mRNA, so mRNA that is going to be converted into polypeptide with the help of a codon, and that codon is what? It is a segment of 3 nucleotide that code for an amino acid. So actually this code or the language is nothing but it is called genetic code, right? This language is nothing but it is called the genetic code. So we will focus how exactly this genetic code, what are these genetic codes and how the language is transcribed. So let's focus on this. So as of now, what we studied in the previous session, we studied about how the transcriptions and the replication take place. Now this is just a recall of the previous session, that what we studied. So you can see the replication process. Like here you can see this is the DNA double stranded, and here you can see this is the sense strand. So now the new mRNA polymerase, you can see the mRNA is synthesized. So this is mRNA, or simply we can say the RNA synthesized, and this RNA is now used for the translation process. So before that there is a genetic code. Now what is this genetic codes? So let's focus on first of all what is genetic code. So as we of now we are very much aware about that there is an intimate connection between gene and the synthesis of polypeptides, or we can say the enzymes. Polypeptide, now you are very much clear about the polypeptide, that is a different amino acid is a acid. So we can say this is one polypeptide, right? So 10 amino acid. So in the modern terminology, a gene refer to the cistron. You know cistron very well, right? So cistron is made up of a large number of nucleotide. Cistron is made up of large number of nucleotide, and the arrangement of nucleotide or their nitrogen bases is connected with the synthesis of protein. By whom? By influencing the incorporation of amino acid in them, right? So that means of, if we simply define genetic code, it is what? The relationship between the sequence of amino acid in the polypeptide chain. Sequence of amino acid in a polypeptide chain, and the nucleotide sequence of DNA or mRNA is called genetic code, right? What these are called? Genetic codes. Now there is a one problem, right? DNA contain only 4 type of nitrogen bases. Yes or no? Who is going to tell me that 4 nitrogen bases? Yes. Okay. Okay. Adenine. Okay. Sargana, guanine. Okay. Thymine and uracil. Cytosine. So can we say it's 5? Yes. If the question is asked about the DNA, then only 4. And in RNA, so thymine is replaced by, right? Yes. That means of this genetic code, DNA contain only 4 type of nitrogen bases or the nucleotide, while the number of amino acid is 20. That means of amino acid number is total 21, but we have only 4 nitrogen bases. So it was, therefore there are some scientists who, hypothesized, or we can say they formulated, or they assumed, right? So what are those scientists who hypothesized? They are nothing but, let's see the name of the scientist, the very first one, that is called the George Gamow. It's George Gamow who hypothesized that since there are only 4 nitrogen bases, and if they must code for 20 amino acid, the code should constitute a combination of bases. What is the combination of bases? That is AUG, AUGC, or we can say ATGC, right? A combination of bases means, that means of ATGC. It should be always in the combination of ATGC. To code for all the 20 amino acid, the code should be made up of 3 nucleotides. Now, 3 nucleotides, that means of if it is 4, so how it can be? Like, so it was first time just hypothesized by this George Gamow. Then later on, one of the famous scientists, before that there is some other scientists also work, but this is what the Khurana, okay, in the year of 1964, Khurana developed a chemical method to synthesize RNA molecule with a defined combination of bases. Okay, it was Har Gobind Singh Khurana. You have to please keep this mind regarding the genetic code. Har Gobind Singh Khurana, who developed a chemical method to synthesize RNA molecules with a defined combination of bases. Now, then there is Marshall, a cell-free system for protein synthesis is finally developed, finally helped the code to be the deciphered. That means of what should be the code that, this Marshall Nirenberg worked hardly to synthesize the to give a actual hypothesized of the genetic code. Later on, it's the Severo Occhia who discovered polypeptide phosphorylase enzymes. Polypeptide phosphorylase enzymes which polymerize ribonucleotide to form RNA without any DNA templates. So it was Severo Occhia. Now, so finally they formulated a genetic code. Now, what is genetic code? Now let's focus on simple introductions of genetic code. So you can see here the genetic code. Slowly all the codes, the codon. I have told you about the codon. What is codon? What I said about the codon? Codon is what? It is a sequence of 3 nucleotide that code for one amino acid. What is codon? It is the sequence of what? 3 nucleotide, 3 nucleotide, nucleotide or code for one amino code for amino acids. Clear? So that means of if suppose there is mRNA like here, this is the mRNA strand. Okay, suppose this is the mRNA strand. Okay, and on this mRNA strand there is a code like I can say A U, then again U, there is C, CAC, then GGA. You know very well what is G, what is C, what is A. Yes or no? Or shall I call by name of amino acid? Or you know very well what is C, what is A, what is G? U, G and C. Okay, so just imagine this is what? mRNA. Yes? So what is codon? The sequence of 3 nucleotide. Now what is 3 nucleotide? So in this you can see initial 3 letter is a 3 nucleotide. Adenine, uracil, uracil is a 3 nucleotide, right? So this is code for what? This is one codon. This is one codon. Okay. Similarly, you can make a triple codon like this. So always triplicate. It should always be triplicate. So this is what triplicate. This means these are the triplicate codon, and it should be specified by a particular name. So see here, AUU. You can see just you can go in this chart and find out what is AUU. So you can just go A, then UU. What is AUU? Can you able to find the AUU? Yes, you can see in this codon, this is AUU, and this stands for what? Isoleucine, right? So this is code for isoleucine. So I can write here this particular amino acid is code for isoleucine. Isoleucine. Similarly, this CAC. Where is CAC? So you have to focus on this code C, then A. So here you can see CAC. So CAC. Yes, it's here. Yes. Yes or no? And what is the name of this amino acid? HIS. What is HIS? It's a histidine. Okay, it's a histidine. So I can again write here. I can go here, take a square, circle, and I can write it here as a histidine. Similarly, GGA. Now you have to go in the first letter, that is G, in the second letter, that is G. Yes, G, and then So first you have to focus on how you will go in this code. So you refer this is G, then again you refer second letter refer that is G, third letter is what? A, so you can refer A. Where is A? So in this GGA, so here is A. So you can just focus GGA. Now you will got, right? This is what? What is the amino acid? This is called GLY for glycine. Okay, this is code for glycine. So again I can go here, take a circle, and I can write here, this is glycine. Similarly, you can refer the another code like UGC. How you will refer UGC? Again, you have to go in U. So U is over here in the first letter, U is over here, then you can go in G, so this is second letter, then C, that means of it should be here. So UGC means this is, and the what is the code of this? Cysteine, right? As and also mentioned in that. So this is what? Cysteine, right? So this is what? mRNA, and this is what? This sequence is called polypeptide. This is a polypeptide. Clear? This is what? A polypeptide. So that is what mentioned over here, that slowly all the codes were deciphered and the checkerboard for genetic code was prepared. So this is a checkerboard method of genetic codes to identify the particular amino acid. Okay. Now, so there are total how many amino acids? So total there are 20 types of amino acid which is involved in the translation. Total 20 type of amino acid which is involved in the translations. Now you have to remember each and every amino acid. So only 20 amino acids. So it is very easy for us to remember, right? So it is what this. The first one is a alanine, arginine, asparagine, then aspartic acid, followed by cysteine, glutamine, glutamic acid, then glycine. So this is total 8 number. Then there is a histidine, which is referred by a short code that is called HIS. Histidine, it should be the capital. Actually, it should be like this, HIS. This is like ISO. So that is isoleucine. Okay, it should be like this only. This is leucine. So LEU, that is called leucine. Lysine, LYS, capital LYS. So capital initial letter is capital and then last two letters. That is in small. So that is what the shortcut of the amino acid. Okay. Similarly, methionine. So methionine is referred as a MET, that is methionine. Then phenylalanine, PHE, capital P, then HE, phenylalanine. Proline, serine, threonine. This is total 17. The another one is a tryptophan, tyrosine, and valine. Okay. So these are 20 different amino acid. Is this clear? Okay. Now, one of the important things which we need to remember from this checkerboard of genetic code was three important things. One is AUG. AUG. You can see here it is mentioned over here, right? AUG. Another one is UAA, UAG, and UGA. These three is called stop codon. Stop codon. And this AUG is called the start codon. This is a start codon and this is a stop codon. That you have to remember. Okay. This is something important that will help you in the genetic, in the translation, that what are the sequence of amino acid and where the reactions start and how the reactions stop. So because of this sequence, AUG reaction is going to start, right? And because of the reaction, because of the stop codon, that is UAA or UAG or UGA, the reaction, that is translation, is going to stop. So that you should know that which code is referred for start codon and which code is referred for stop codon, because this is important. So you have to just remember. Okay. Yes, all of you noted down what is stop codon and what is start codon. Clear now? Okay. Now let's focus on the salient features of genetic code. Now, what are the important characteristics, or we can say the silent features of genetic code? So there are important salient features of the genetic code is, first of all, the genetic code be triplet. Now, why it is triplet in nature? Let's first of all focus on. So as of now, we studied that the three adjacent nitrogen bases constitute a codon which specify the placement of one amino acid in the polypeptide. So it should be triplet in codon. Okay, it should always be triplet. Okay, then so triplet codon means three codon do not code for, as we know that the code is triplet because made of three adjacent nitrogen bases. A triplet codon specifies the placement of one amino acid in the polypeptide chain. So there are total, as we can say, that 61 codon code for 20 amino acid, right? So 3 codon do not code for any amino acid, and the one code that stop the codon, right? So you can just count in this total how many it is. Yes. So just count it. How in total? How many total codon in this checkerboard method? 64. And out of the 64, as I told you, 3 codes do not code for any amino acid. 3 codes do not code for any amino acid. So can you tell me what are those 3 codes? Yes, it's absolutely right. As I told you, that is stop codon, because you can see in this checkerboard method only, there is no written that the stop codon is a kind of amino acid, right? It is not mentioned. It is just mentioned as a stop codon. So it having no codes, right? So UAA, UAG and UGA, these are 3 stop codon, and that's why this codon having no any amino acid code, and that's why we have total 21 codes. So out of 64, 3 do not code for any amino acid, and they are called the stop codon, right? Yes. Now the second one, second option, second salient features, unambiguous and specific. Now what is mean of ambiguous? Unambiguous means specific, or we can say the code is ambiguous, means one code is specify only one amino acid. Means if there is UAA, or we can say UAG, AUG is code for methionine, then all the in that 64 codon, if there is AUG, it is always code for methionine, right? That means of there is no any conspiration, that like we can say there is no any uh, unambiguous, or we can say specific. So one code is very specifically for one amino acid. There is no any other amino acid for the same code, right? Is this clear? Second point and first point. I want response from everyone. Yes, please respond, all of you. Ragul, Sasidharan, Jason. Why are you not responding, Jason? Ragul, any doubt? Kirtini, Prasanti. Okay, okay, great. Sasidharan, got it. Okay, now let's move ahead. So, first salient features of genetic code is triplet, unambiguous and specific. Now you know the meaning of unambiguous and specific. Now let's move ahead and try to understand the next, degenerate. Now what is the meaning of degenerate over here? So degenerate means what? That once the code, or simply we can say, yes, so the code is degenerate means all the amino acid except tryptophan and methionine are coded by more than one codon. So let's again observe that checkerboard method of genetic code. So in this checkerboard, you can see that except tryptophan and methionine. Now let's move and try to understand in that checkerboard. Yes, here. Tryptophan and methionine. This is a methionine over here. Yes or no? And here you can see the tryptophan. Yes or no? Yes. So methionine except tryptophan and methionine, other amino acid, all amino acid are coded by more than one code. So you can see, just check that this is PHE. Now what is PHE? It's a phenylalanine. So you can see it is code for this one also. Then you can see the phenylalanine somewhere. See how many codes are there? Triple U or UUC, right? That means of phenylalanine can be referred as a triple U also. It can be referred as a UUC also, right? Even leucine. So you can see UAA, UUG, even CUU.
Sir, you
Then C.
circled tyrosine
Yes,
instead of tryptophan.
yes, yes.
Instead of tryptophan, you circled tyrosine in the checkerboard.
Let me check. Tyrosine. Yes, yes. Yeah, it's here, right? Extremely sorry. Yes, because of two codes are there, right? Yes, yes, I got it. Yes. So here you can see this is a tryptophan. So tryptophan having only one codon and the methionine having only one codon. And except all these codon are more than one, they code more than one amino acid. Is this clear? So you have to also remember, this is also important point, that methionine and, let me write here so it will help you to understand. Yes, degenerate, that means of except, let me write here, methionine, that I can write as a met, and another one that is called tryptophan, right? So this two codon is not going to be repeated, right? But all amino acid code are going to be repeated. So it's a degenerate. Methionine and tryptophan is not going to regenerate, but except these two, all codes are regenerate, or we can say degenerate. Another one is no punctuation. That means of while writing this triplet genetic code, it should be in same sequence without any punctuation, without any comma. Like suppose if I'm writing GUG, that means of there should not be any comma, any punctuations in between this 3-letter code. So it should be always written in the form of GUG, like this only. And that's why it is mentioned over here, no punctuations, no comma. Clear? So codon, yes, is this clear? So it should be not, there should not be a comma, pause or any kind of overlapping in between the triplet codon. It should not be overlapping. What is mean of overlapping? So here suppose this is a code like GUG, then CAU. It should not be overlap. It is always in a triplet codon like this. Yes, so no punctuation in between. So this is third important salient features of genetic code. Now the fifth one is universal, nearly universal. Now nearly universal means what? The genetic code are universal. As you know that a codon specifies the same amino acid from a virus to a plant to a human beings. Means from any type of organism if you are referring, you can just consider that this amino acid is universal. It is referred everywhere as the same name, that is if there is a methionine for start codon, so it is referred for a small organism also, it is referred for higher organism also, it is same, it is not going to be referred by different name, and that's why it is universal, right? So some exceptions for this rule are present in the mitochondrial codon and even in the some protozoans, but that can be exceptional. So you just remember in the mitochondrial codon and even the in a protozoans codon there is exceptional, but it is referred, genetic code is universal and it is referred everywhere as the same, from the smallest microorganism to the highest tree, or we can say the plant, any plants, right? Now codon AUG, now this is something important. Codon AUG has a dual functions, means it having two type of functions. Codon AUG, what is AUG code for? Yes, I want to know from Sasidaran, what is AUG code for? AUG code for what?
Sir, it is the start codon and it code for methionine, sir.
Yes, absolutely right. Okay, so it's a start codon and it code for methionine, right? Oh, it's already written. AUG is code for methionine and also act as a okay. So as I told you that genetic code is universal and this codon has a dual functions. Why it has a dual function? Because it code for methionine amino acid and also act as an initiator codon. Right? They have a methionine codon also and work as a initiators also. Means to start the translation, this work as a initiator. So it having the amino acid code also and also act as initiator, and that's what is called the dual function. AUG has a dual functions, right? Is this clear? Okay, one more important things, and that is not always, but rarely, I'm saying rarely, GUG, this is rarely, I can write here so it will be more convenient for you. It's a rarely, yes, I'm writing rarely GUG, or simply you know GUG is what it is, a valin code, act as an initiator codon. It is valin code, act as a initiator codon. Not always, rarely I'm saying. So this is also you have to remember. This is also important. Okay. So total how many salient feature we studied till now? Total 6. Yes. The very first one is triplet codon. Then there is a unambiguous and specific. This is second salient features. Then degenerate. This is third salient features. Then there is a no punctuations. This is the fourth salient features. Nearly universal, and the AUG is code for methionine and also act as an initiator codon. So it have a dual functions, right? So these are the salient features of genetic code. Now let's move ahead. Okay. Now I would like to tell you something regarding the mutations also in this genetic code. So if insertion or deletion of one or more bases, you know that what is mean of insertions and deletion. Insertion means addition and deletion. So if any additions or deletion in the nitrogen bases change the reading frame of mRNA from the point of insertions or deletion, then it is come under the frame shift insertions or deletion mutations. That can also be possible, right? And even insertions or deletion of 3 or its multiple nitrogen bases, then this insert and delete one or more multiple codon, and hence because of the deletions one or multiple amino acid, this cause the reading frame to remain unaltered from that point onwards. So it is going to be unaltered after that if there is a mutations in that mRNA code. So that can be also be possibilities in that. But overall, there are total 6 salient features of genetic code. Now, hope this is clear to all of you. Yes? Yes. Sarjana, you are talking something?
Sir, actually, I have a doubt, sir.
Yes, please ask it out.
Sir, you said it is nearly universal, no, sir.
Yes.
Like, did you mention, like, is there any exception, sir?
See, I have told you that the genetic codes are nearly universal. That means of what? A codon specifies the same amino acid from virus to the plant. What is mean of universal? That means of if you are studying on virus, then virus also having the same amino acid, or that amino acid can also be referred for plant also, that amino acid can also be referred for human, means all type of organisms. If you are studying at the amino acid level, that means of all these amino acid code should be universal. So some exceptions to these rules are present in the mitochondrial code and the protozoans, because they have something different codon. So that's why mitochondrial codon and the protozoans have some different, and that's why they are referred as, some exceptions are there.
Okay, sir.
Hmm? So
Yes, sir.
keep in mind mitochondrial codons and the protozoans. These are some exceptions in that universal code of salient features of genetic codes.
Okay.
Clear?
Yes, sir.
Okay. Any other doubt? No. Okay. Let's move with the next. Now, as of now, we studied about the genetic code, and we are very much clear about it, that what is genetic code, right? Yes and no? Come on, respond it fast. I want response from everyone. Then I will move with the next concept, that is translation. Okay. Well, Now let's move with the translation. Okay, so translation, simply it is mean of protein synthesis, right? Yes, okay, that's great. So let's move ahead and try to understand. Now you know translation means what? When RNA translate it form a protein. So that means how we actually translation means what? We are going to study about the protein synthesis, how the protein is synthesis in a particular organisms, right? So it consists of, they have a special machineries which is called the translation machineries. And what are those translation machineries? So basically those translation machineries are, you can see here. So this is just a definition that translation of the process, translation is the process of converting or translating the sequence of an mRNA into a sequence of amino acid by peptide bond to form a polypeptide chain. So here you can see this is RNA, and this RNA means mRNA, and this mRNA, when it translate, it form a protein, okay, with the help of ribosomes. Okay, so it form a protein. So this is what the polypeptide chain we can say, right? So what are the important machineries required for the translations? So for translation, it consists of ribosomes. That means of very first tools required for translation is ribosomes. Means for initial translation we required ribosomes, we required amino acids, we required mRNA. You know mRNA, where you will get mRNA from the transcription, that mRNA, tRNA, and the amino acyl tRNA synthetase. Clear? These are the 5 important, let me write so it will be more clear. The first things we require, I think so it is already there, it is ribosomes, the first one. Then we require amino acid, then we required mRNA, we also require tRNA, this is the fourth one, and amino acyl tRNA synthetase. So these are total 5 important machineries, or we can say tools, required for translation. Clear? Now mRNA is a messenger RNA. Okay, we required a ribosomes. So initiation factors, elongation factor, and release factor is also required. These 3 important factors. Then there is a amino acyl T synthetase. Then we require tRNA, then transfer RNA, and ribosomal RNA. So let's focus one by one. The very first, we required ribosomes. Now in that translation machineries, mRNA function as a template having genetic informations. Means in the first, this mRNA is what? This is a strand which having the genetic information. This is referred as a mRNA function as a templates for what? For having genetic information. Having genetic information. Now the another one, ribosome is the site for protein synthesis. Where is the ribosomes? You can see here. So this is what? Site for protein synthesis. This is site for protein synthesis. Then tRNA. Why we required tRNA in the translation? So tRNA bring the desired amino acid. tRNA, what is the function of tRNA? So tRNA bring the desired, so let me write here, it brings the desired tRNA brings the desired DNA, sorry, not DNA, desired amino acid. Brings the desired. Can I write AA? It is very much clear. What is AA? It's amino acid. Or you want me to write full form? Okay, let me write full form. So it is amino acid. So tRNA bring the desired amino acid, and also this tRNA read the genetic information and place the amino acid at proper place. That means of whatever the desired amino acid brought by this tRNA, it is going to be placed according to their proper placement. So basically the places of amino acid is proper. So it is also going to placed, like suppose if you are reading with some institute, so that institute is going to be to do a placement. Similarly, this tRNA is also providing placement. Okay, so I can write here that uh, uh, this tRNA bring the desired amino acid. Also, it read the genetic informations and place the amino acid at proper place. So it basically it place the amino acid at proper place. Is this clear? Total 3 important function. Very first, what is the function of tRNA? tRNA bring the desired amino acid. Very first. Second, this tRNA is going to read the genetic information. And third, it also places the amino acid at proper places. So simply, this is the first, this is second one, and there is no place for writing the third. Okay, I can write here. The third important function by the tRNA, that is read genetic information. Genetic information for okay. So it basically read the genetic information. That's all. Okay, so bring the desired amino acid, read the genetic information, and place the amino acid at the proper place. That is the function of tRNA. Yes, in this machineries. Now the another requirement, let's focus. The next one, that is RNAs are formed over DNA during transcriptions, while the protein synthesis occurs in the cytoplasm over the ribosomes. That means of you have already studied that uh, transcription in prokaryote and transcription in eukaryote in the last session. Yes or no? Just tell me. Okay. So who is going to tell me that in prokaryote and eukaryote, transcription in basically take place in which type of organelles, in prokaryote and in eukaryote? No idea. Come on, tell me fast. Yes. responding. Why? Am I audible to all of you? Am I audible? Is my question is clear to everyone? What I asked? Yes. In cytoplasm, in prokaryote, in nucleus, in eukaryote. Okay. That is what I'm asking. Very simple questions. Right? So I told you in the last session that transcription take place in prokaryote in the cytoplasm, because there is no any prominent nucleus, right, in the prokaryotic organisms. But in the eukaryote having proper nucleus. So basically this transcription take place in nucleus in the eukaryote, right? So that means of this entire, right, these RNAs are formed over DNA during transcriptions, while protein basically, that means of RNA, that DNA converted into RNA, that is called transcription, which is take place in the nucleus, right, and it translated into the cytoplasm. So the two are separated both the space and time, so it prevents the intermixing of raw materials, and it also protects DNA from the respiratory enzymes and ribosomal machineries from the nuclease. So this is what the reason behind it. So these are all the important machineries, or we can say translation tools required for translations. Now let's focus one by one the structure of each components. So very first there is a ribosomes. Now what is ribosomes? Now we have to focus what is ribosomes. Now so you know very well that protein synthesis occurs over the ribosomes, and these ribosomes are therefore called the protein factories. What ribosome is called? The protein factories. And each ribosome has two unequal parts. You can see here these two unequal parts is what? One is called the small subunit and the another one is called large subunit. So can you see here the small and the large subunit? No? Yes? Yes. So here you can see in this diagram, this is the large subunits and this is the smaller subunits. So larger means it's little bigger in size and small means little smaller in size. That is the difference between the large and the small subunits. Now you can see this, the large subunit of ribosome has a grooves, for pushing out the newly formed polypeptide and protecting the same from the cellular enzymes. That means of large subunit having a special character. That means of in this large subunit they has a grooves, and this groove, why these grooves are present in that? For pushing out the newly formed polypeptide. So newly formed means what? After the translation the new polypeptide are formed and that are going to be pushed forward, right? So it is basically that grooves basically help to pushing out the newly formed polypeptide and protecting the same from the cellular enzymes. This is what the characteristics of large subunit. Similarly, you can focus on the smaller subunits. So where is the smaller subunits? So here you can see the smaller subunits. So this is smaller subunit that fit over the large subunit like a cup but leaves a tunnel for the RNA. That means of it is hold like this. Suppose You can just focus on the my screen, my what we can say image. Like here you can see. So suppose this is the large subunit, this small subunit. So these are in such a way that it is going to fit inside like this, but there is a gap. Through that gap, RNA can easily pass and translate. That gap should be there. So they are going to fit in such a way that the RNA can easily translate the proteins like they are fittings. Okay, so this is what the formation in this ribosomes. So basically smaller subunit fit over the large one like a cup, but leaves a small tunnel for the RNA. So there is a minute hole in between these two subunits. So in that tunnels, that is what simply you can refer as the tunnels. So in that tunnels, like how you know the train passing through the tunnels, so it is like that only. Okay, so there is a minute tunnel, so the mRNA can easily pass out. Now the two subunits, when they are come together only at the time of protein formation, means at the initial stage they all are separate. That means both are separated at their own place, but at the time of protein formations they are coming together. So when these two subunits are reunite at the time of protein synthesis. Now, so when they come together, this phenomenon are referred as associations. Means when the larger subunit is smaller subunits, when both are come together, you can see in the upper diagram. So this one is the upper diagram. So this is the large subunit, is smaller subunits. When both are coming together, they form a like this structures. So this is what called associations. So here you can see this is the association of small and larger subunits. Now here magnesium is essential for it. That means of in this particular magnesium is very important. That is Mg+2. Okay, so soon after the completion of protein synthesis, these subunits get boom. So what happened? That means of they're going to separate. Okay, they are going to, we can say they are joined, and when the protein synthesis is completed, they blast and they separated to different their own path. So they dissociated. So they associated, and once the protein synthesized, later on they get dissociated, means different. So the phenomenon of separating these two, suppose these are separated, so can I call it as a dissociations? Yes, I can refer as a dissociation, right? Clear? So it's associations and dissociations. So when they are separated, it is a dissociations. When they are united, it is associations. Is this clear to everyone about the ribosome, the characteristics of ribosome, the larger subunits, the smaller subunits? Yes. Okay. Now, one more interesting things in this ribosomes. There's a different type of ribosomes in the prokaryote and the eukaryote. So you can see in the eukaryote, the total size of the ribosomes is 80S. S stands for Swedberg unit. It is a unit, a Swedberg unit. So that's why it is written as a 80S. So S stands for Swedberg unit. Okay. Now here you can see when this RNA is at the dissociation, this ribosome at a dissociated stage, the larger subunit having a 60S and the smaller subunit having a 50S. But when they are unite, they form associations and it becomes the size of 80S. Now again, the question in your mind that if there is a 40S and 60S, it should be 100S, right? Then why it is 80S? Yes or no? Okay, so let me tell you, it is a it is a sedimentation rate. It is based on the sedimentation rate. So that's why, because of the sedimentation, that means of the settlement of molecules, like two different subunits, they are going to be, combined. So they have adjustment settlement. So sedimentation unit, based on that sedimentations, it is the size which is ATS. So don't think uh, much about it. It is 80, and at the time of dissociation they have a 60 and 40s. So it is based on the sedimentations. It is, the size is reduced, so it become a ATS. So 60 and 40 is a large and smaller subunits, and when they are associated, it become a ATS. Similarly in a prokaryote also, but slightly different. Like here you can see, in the prokaryote, the larger subunit is referred as a 50s, the smaller subunit referred as a 30s, and when these two dissociated ribosomes, when they are associated, they form a 70s ribosomes. Here, now along with that, you have to also remember the important things, that is 60s means what? They have 28 RNAs, 5.8 sRNAs, 5 sRNAs, and 50 proteins. In case of 40s, it is having 18 sRNA and 33 proteins. So this count is also important. Okay, so I think so this is important, some marking here, because 2 or 3 questions from the previous year, you can just check that you will get your answer in this chart only. Okay, so this is important, so you can just refer it. Now here in case of prokaryote, you can see that is 23 sRNAs, then 5 sRNAs, then 32 proteins. Similarly in here, that 16 sRNAs, 21 proteins, right? So these are the complete informations about the ribosomes, which is called the protein factories. This is a protein factory. Okay, you know the factory, any product if manufactured in the factory, so it initially they require different kinds of raw materials, and when this raw material, when we put into the factory, it release some new product. Similarly here, this 2 ribosome is work as a factory, and when the mRNA, when it pass into the tunnel, it form a proteins which is coming out, right? So yes, let's move ahead and try to understand the remaining component. So very first component, that is ribosomes. I hope that everyone clear about it. Yes, or still anyone have a doubt on this ribosome, then please ask me. Yes, anyone have a doubt? Shall I move ahead? Okay, now come to the another one, that is amino acid, and this is already well. So 20 different amino acid occurs in the cellular pools, which are arranged in a specific sequence in the protein to impart its specific nature. Like what kind of nature? Like there are 20 amino acid, all these 20 amino acids having the different polar and non-polar groups. So you can see here, we have out of this 20 amino acid, we have total 8 amino acid as a non-polar group. So you can see this is the non-polar group: alanine, valine, leucine, isoleucine, proline, phenylalanine, then tryptophan, methionine. These all are the non-polar group. Then there is a polar group that having the glycine, serine, threonine, then there is a cysteine, then tyrosine, then asparagine, and glutamine. Then out of this, there is also having some are negatively charged, some are positively charged. So negatively charged are aspartic acid, then glutamic acid. In case of positively charged, lysine, arginine, and histidine are the positive amino acid. So these are the different 20 amino acid having, they are all categorized based on their polar and non-polar, negatively and positively charged group. Now come to the mRNA. Now you know very well about the mRNA. So the messenger RNA or mRNA, which constitute about 2 to 5% total RNA, and consist of a codon, means they have a codon information from the DNA, and they take part into the translation of by binding amino acid in a particular sequence to synthesis a protein. So this is the pre-mRNA after the RNA polymerizations. Now this is after transcriptions. Now this is the complete mRNA strand. So this mRNA strand is going to pass in the tunnel of ribosomes, okay, at the time of protein synthesis. Now we also required tRNA. So you know the role of tRNA. What is role of tRNA, by the way? I've told you the role of tRNA in the protein synthesis. So I just wanted to know from you. Yes. I already wrote on the screen that what is the function of tRNA, right? Read the genetic information, okay? Okay. Read genetic code, okay? Both responding the same. Bring desired amino acid. Wow, that's great. Bring the desired amino acid, okay? One more character is there, I think so, right? Yes, place the amino acid at the proper particular place, so we can say the proper place, right? So yes, tRNA playing a very major role for, so you can see here they are transfer tRNA. You know the meaning of tRNA, it's a transfer, or simply we can say soluble RNA which pick up the particular amino acid in the process called, that is called the charging. That means of when this tRNA pick up a particular amino acid, that means of it is going to be charged. So that process is called, when it pick up that amino acid, particular amino acid, when it pick up, it is called charging. What it is called? Charging. So the charged tRNA take the same to mRNA over particular codon which correspond to their anticodon. So a tRNA takes the takes the name to mRNA over particular codon corresponding to their anticodon. So tRNA can pick up only a specific amino acid. On the amino acid can be specified by near about 2 to 6 tRNA. So each tRNA has an area for coming in a contact with the ribosomes. What is that area? That is T psi C. And where is T psi C? So I have already told you, and we have already discussed about the tRNA in the previous session. Yes or no? Yes? We have already discussed what is rRNA, what is mRNA, what is tRNA in the previous session in the cleaver leaf model. Are you remembering that what we studied in the last session? Yes. Okay. So in that cleaver leaf model, what happened? The amino acid, you know that, hello?
Sir,
Yes.
can you explain tRNA again?
Okay. Yes, I can explain. No problem. See here the diagram, I think so diagram is almost same, right? Yes or no?
Yes, sir.
Hello.
Yes, sir.
Okay. Okay, so just pay attention on this diagram. Now here you can see the tRNA. I told you in the previous session that tRNA is the adapter molecule. Why it is called adapter molecule? Because the codon of amino acid, the codon of mRNA are not recognized by the amino acid due to their lack of structural speciality to read the amino code uniquely. So that's why there is a Francis Crick postulate the presence of adapter molecule, and that would one hand read the code and one other hand could bind to the specific amino acid. So this adapter molecule is called the tRNA, or simply we can say the transfer RNA, or it is also referred as a sRNA. Means tRNA is also referred as a sRNA. tRNA, or you can say sRNA, small s RNA. sRNA for soluble ribonucleic acid, or tRNA transfer ribonucleic acid, you can, both are the same. Okay, now here the tRNA which consists of 15% total RNA, as I told you, right? So that means of out of 100% RNA, 15% is a tRNA, and it has a two-dimensional cleaver leaf like structure. Like you can see here, this is what the two-dimensional clover leaf model. This is clover leaf model. Clover leaf model. Clear? Now this clover leaf model structural and the three-dimensional L-shaped structure. That means of in the this is a two-dimensional structure. This is 2D, and in case of 3D, it is referred as a L-shaped structure. L-shaped. So in case of 3D, it is L-shaped. In case of 2D, it is a clover leaf model. So mostly question asked on clover leaf model. So what is this clover leaf model? So here you can see the tRNA bind with a particular amino acid at their amino acid acceptor end. What is the amino acid acceptor end? It is CCA. Can you able to find the CCA over here? Yes, it is over here, right? This is the acceptor amino acid. Yes or no? So that means of the tRNA bind with a particular amino acid at their amino acid acceptor, and that acceptor end is with CCA, which process that called the charging of tRNA. That means of when it bind, there is charging, right? When it pick up the amino, as I told you that tRNA pick up a specific amino acid. When it pick up the specific amino acid, the process called charging, right? So here it is attached with the CCA, and that process is called the charging of tRNA. Charging of tRNA. Now, so what happened? The charged tRNA takes into amino acid takes it to amino acid mRNA over its specific codon, where hydrogen bonds are formed between its anticodon. So where is anticodon? Which is present on anticodon loop. So you can see here, it is present anticodon loop, right? So here is a anticodon loop which is present. So this anticodon loop that correspond to the codon of mRNA on the mRNA. Now the tRNA molecule also has an area for coming a contact with the ribosomes, and that area is referred as a T-Cy-C loop. This is the contact of what? This is the area for coming in contact with the ribosomes. But that means of if there is a tRNA, so where this tRNA will go? It is going to contact with the ribosomes. So at what point they are going to contact? With the help of this anticodon loop. So you can see the anticodon loop where it is present at the base. So this is anticodon loop, so it is going to contact. So this is mRNA strand, this is mRNA strand, and this tRNA going to contact with this anticodon loop. So now there is a connection. So this is the anticodon, so it is going to contact with this mRNA, right? Now this tRNA molecule also has an area, that is T-Cy loop, and the enzyme amino acyl tRNA synthetase, and that is called DHU loop. So here is a DHU loop that is going to be attached. So DHU loop is there. That what is mean of DHU loop? I have told you what is DHU. Yes, you all forget? Just tell me fast. What is the DHU loop? Yes. Okay, let me check how many you respond. No one responded yet. Why? is this DHU loop? It is a dihydrouridine. I have told you in the previous session. Dihydrouridine. Right, so it is a dihydrouridine loop. Okay, so it is present on tRNA. And what is the main function of this DHU? So DHU is the enzyme amino acyl tRNA. It is going to be synthesized at DHU and their variable arms. So where is the variable arms? Which is present over here. So this is what the important that you need to remember, that in the clover leaf model, what is the structure of tRNA, that you should be very clear with the structure. Now I have also told you about the adapter molecule, why it is called adapter molecules. Yes, is this clear? And Annie, Anmol Deep, yes, it's Anmol Deep. Yes, Anmol Deep, clear? Okay, okay, great. Okay, now the next reactions, like amino acyl synthetase. Now, what is the amino acyl tRNA synthetase? The enzyme amino acyl tRNA synthetase combine an amino acid to its tRNA. So there is a specific enzyme for each amino acid, and that's why you can see this is what, this is the tRNA, this is a anticodon. So this and here is a end, right? So when this amino acid, when is it pick up, it contact with this band, what happened? This process is called it as a amino acyl tRNA synthetase. Clear? That means of, as I told you, that in that loop, in this tRNA outer molecule, you can see there is a charging process. I told you what is charging. The tRNA bind with a particular amino acid at their amino acid acceptor end. This process is called charging tRNA. So charging tRNA means what? Here is the amino acyl tRNA synthetase. So with the help of this enzyme, it is going to be charged. Now it become activated. So once it is activated, now it is going to bind with the mRNA. So this is what overall enzyme, this is the enzymes which is required to become activated, right? So this is the what, this is the initial stage of requirement of protein translations. Now let's understand the mechanism of translations. What are now, we are very much clear that for translations, what are the basic ingredient required for translations, or we can say basic machinery tools required for translations. Yes, now let's understand the process. That means of, in the factory there are various process, like first of all checking the material whether it is good quality or bad quality. Yes, then there is a different process of designing, then coloring, then everything, like all in a proper sequence. So here for translation, it is also having a proper sequence. That means of which tools is going to bind first, then what reaction will come, then the next bind, like that. So the very first mechanism of translation is a activation of amino acid. That means of amino acid should be activate. So how it is going to be activate? So very first, for activation of amino acid, in the presence of ATP and magnesium, what? In the presence of ATP, you know ATP, yes. So in the presence of ATP and magnesium ion, So in the presence of ATP and magnesium, an amino acid combined with its specific tRNA synthetase to form amino acyl adenylate enzyme complex. So here you can see this is the activation of amino acid. Okay. Now charging of amino acylation of tRNA, charging or amino acyl. So how it is going to be charged? So the next step is charging. So how it is charged? You can see here. This is what the ribosome. This is amino acid. When amino acid come in a contact with the tRNA, it is going to be charged. That means you can see the ATP is also there, magnesium is also there, and the phosphate group which is releasing that ATP. Can you able to see the ATP written over here, that yellow one, ATP? I know this is a little smaller, but I think so it visible to me. So hope it is also visible to you also. Yes. So this is what ATP. So how the charging take place, that charging of charging or the amino acylation of tRNA take place. So basically the amino acyl adenylate enzyme complex, when it react with the tRNA specific for the amino acid, it produce amino acyl tRNA complex, and this amino acyl tRNA complex is called the charging tRNA. So after ATP absorptions, it is going to be fully charged. So now it is the enzymes and the AMP are released, because you know that the TRP, when it release ATP, then you can see the phosphate group and all, they are going to be tRNA and all, right? So this is what the charging of amino acyl. Now the reaction start here, that is initiation. Now how the initiation take place? So you can see the mRNA attach itself to the smaller subunits of ribosomes. Where is the ribosomes? So you can see here, this is a large subunit, this is smaller subunits. Yes or no? So this large subunit is smaller subunit. When it attach itself, then there is what? The attachment is in such a way that the initiation codon of mRNA comes to lies at the P site. Now where is in this particular, where is the P site, A site, and that is important. So let me tell you what is P site, A site. Yes. So here in this diagram, is this visible to you? Yes. Now this is what the ribosomes. This is the smaller, this is the larger ribosomes. I can write it as a large subunit. It is written as larger ribosomes, and this one is a smaller ribosome subunit, and this is the mRNA. So this is the mRNA. This is, now here you can see in this mRNA, along with the ribosomes bind, you can see there is written as A site, P site, and E site. These three site is important. What is A, P, and E? What is mean of A, P, and E? Let's focus on it. So in this diagram, you can see there are three reactive site. These all three are the reactive site, A, E, and P. Now P site is referred as a peptidal transfer, or we can say the donor site. P site is referred as a peptidal. Can I write over here? Yes, it's a peptidal. Yes, it is a peptidal transfer, or you can refer as a donor site. This is donor site. Okay, this is P site. This P site is jointly contributed by the two ribosome subunit. Okay, that means of half from the larger subunit, half from the smaller subunit. They are jointly compiled, so it is a both are jointly contributed. Then there is a site. A site stands for amino acyl, or we can say the acceptor site. This A for amino acyl, or we can say the acceptor site. So this is donor site, this is acceptor site. Then this A site is situated on the larger subunits of the ribosome. Means this is shared by both ribosomes, and this is only on the larger ribosome subunits. Okay, that means of A is basically fixed in the larger subunits. Then there is another one which is called the E site. So E stand for exit site. So it is a exit site is the part of larger subunit facing the tunnel site. Okay, so this is the exit site. Okay, that means of first of all amino acid or any kind of amino acid, when it come, it bind with the A site, then it reach to the P site, then P is a donor, so it dissociate like in the form of polypeptide chain, and once the protein polypeptide formed, it reach to the E site. E stand for exit site, so tRNA is exit from the RNA from these ribosomes. Clear? So this is the site, okay, that you have to remember. So as I told you, here you can see that the larger subunit and the smaller subunits come together and bind the attachment in such a way that the initiation codon for mRNA comes to the lies at the P site. It required GTP and initiation factors. That means of once it bind the P site, it required GTP and the initiation factors. So in this initiation, you can see during the initiation, the component of the transitions apparatus come together with an mRNA and a tRNA carrying the first amino acid, that is AA1, bind to the start codon AUG. So here you can see in this mRNA, here is a start codon, here is a stop codon. That means of it is going to come over here and it is bind over here. That means of this larger subunits is directly to reach to the start codon site and start binding. It is not going to read from the stop codon. It is start from the start codon. So you can see it is bind. Now here you can see this is P, this is A site, this is P site, and this is E site. Okay, this is the sequence EPA. A site, P site, the tRNA bind with the P site. Why it is bind with the P site? So you can see here, together with the mRNA, a tRNA carrying the first amino acid bind so to start the codon. The next step, that is called the elongation. Now after binding, now this machinery is ready, okay, in the elongation phase. Now you can see in the elongation phase, which is called the polypeptide chain formations. So during this elongations, amino acid are brought to the mRNA by tRNA and are added. So one by one to a growing polypeptide chain. It is going to see here, first one is attached over here, first amino acid, then it reach to the A site, then it start converting like this. So there is continuous chain of polypeptide like this, okay, and it start moving like this. When it reached the stop codon, okay, so Initial stage of initiation is very simple. That means of there is a elongation factors. So charged tRNA reach A site and attach to the mRNA codon next to the initiation codon with the help of its anticodon. It required GTP for elongation factor. The peptide bond is established between the carboxyl group amino acid and then P site. At the P site, an amino acid of the amino group which is attached to the tRNA at A site. Means once it is attached to P site, it reaches to A site and the reaction is catalyzed by an enzyme peptidyl transferase. So transferase means it's going to transfer. So in this process, connection between the tRNA and the amino acid at the P site breaks, and then the ribosomes then translocate along with the mRNA to 5' and to 3' directions, which result in the translocation of A site mRNA codon along with the peptidyl tRNA complex. So that means of A site to P site and the free tRNA at the P site to slip to the E site. So this is what the translocations require for elongation factors. Now after this you can see the next stage is translation, a termination. So initiation, elongations, now the next is a terminations. Now here this is the termination stage. Yes, you all there? Sasidharan, Prasanthi, Kirtini. Now you can see in the termination stage what is happening during this termination. A stop codon in the mRNA is recognized by a protein. That means of you can see there is a stop codon in the mRNA. As you can see in the initial stage also there is a start codon and stop codon is already there in the mRNA, right? So this is going to be read by the tRNA, and so there is a tRNA and there is an attachment of releasing factor is also attached over here. So because of the releasing factor and UG stop codon, now no amino acid is going to be accept. So it is going to be stop the reaction, means amino acid is polypeptide is ready now. Okay, so during the termination the stop codon in the mRNA is recognized by a protein releasing factors, and the translocation apparatus come apart, means they are going to be separated because of these releasing factors. The both apparatus means larger subunits and the smaller subunits get separated and it releases the complete polypeptide. So you can see this is the complete polypeptide which is referred as a protein, right? So this is complete polypeptide, and here this both are dissociate like this. So it get dissociate like larger subunits, smaller subunits, and here there is a mRNA, then there is a tRNA. So this is what the complete process of mechanism of translation. Yes, so initiations, elongations and terminations. Clear? So you know very well the stop codon. So so what can be the stop codon over here? So basically three type of stop codon is can be possible, like UAA, UGA and UAG of mRNA which reach the A site. These stop codons are not recognized by any tRNA. So the P site is hydrolyzed and the completed polypeptide chain is released with the help of releasing factors. So releasing factor is there and it is going to be released. So the ribosome slip off the mRNA chain and dissociate into the smaller subunits and the larger subunits. Now the reaction is completed and the protein is synthesized. So this is what in the case of prokaryote. So prokaryote is very simple. Now there is, so similarly there is in eukaryote also. The little difference is that for the machinery is slightly different. Otherwise in prokaryote and eukaryote basically follow all these three processes, that is initiations, elongations and terminations. Is this clear? Yes, just tell me till now, is this clear to everyone? Okay, now these are some antibiotics which is having a bad impact on the protein translation. So you have to remember the antibiotics which hamper the protein synthesis process. So you have to remember the streptomycin, which inhibit the initiations of translation and cause the misreading. That means of this streptomycin is an antibiotics who can have a bad impact on the protein synthesis, and even it cause a misreading of what, of the mRNA, or we can say the code which is there, that is going to be misreading also. So streptomycin is a kind of inhibitor, we can say it is a inhibitors mechanism. This is, you can refer as an inhibitor. These are all inhibitor chemicals. So streptomycin, tetracycline. What this tetracycline basically work? So it is basically worked on inhibiting the binding of amino acyl tRNA to ribosomes. Then there is a chloramphenicol. So chloramphenicol inhibit the peptidal transferase, so the formation of peptide bonds. And similarly, there is a erythromycin, is kind of another antibiotics who basically inhibit the translocation of mRNA along with the ribosomes. Then there is a neomycin, which inhibit the interaction of tRNA with mRNA. If there is no interaction between the tRNA and mRNA, then imagine what will happen. No protein synthesis will take place, right? So basically, this kind of chemicals is work as an inhibitor to stop the protein translations. So you should also remember that what are the inhibitor who have bad impact on the protein translation. Then there is a puromycin. Puromycin is bind to C terminus of growing peptide chain and cause premature termination of protein polypeptide chain in both prokaryote as well as eukaryotic translation. And there is a rifampicin, and there is a actinomycin. Rifampicin and there is a actinomycin. These two type of antibiotics which is inhibit the RNA synthesis by inhibiting the RNA polymerase. So rifampicin and actinomycin, these are two important inhibitors. So all inhibitor you have to remember, total 7 inhibitors. I think so one or two question asked in, I think so previous, or I think 2020, it's a 17 or 18. In that there is a question that which of the following is a inhibitors for protein translation. So you should remember that which of the, so these are total list of protein translations. So you have to just keep in mind the total important antibiotics which inhibit the translation. Is this clear? Yes. Okay. Now let's move and come to the question assisted practice. Now the question in front of you, in the protein synthesis, which of the following are required for synthesis of charge, required for the synthesis of charged tRNA, means charging of tRNA. What are the important things required? To become a to charge tRNA, what are the essential important molecules required for charging of tRNA? Okay, let me generate the poll. So I'm going to generate the poll and you have to answer in the poll. Yes, ready? So I'm writing the first, just writing first question. First question, and in that option is A, either B or C or D. Now you have only 60 seconds to respond. So within the 60 seconds you have to respond. So just tell me, can I generate the poll right now? Yes? Okay, now the poll is generated to you. Okay, so, oh my God. Why 50-50% response? Some selected answer B, some selected answer D. Why? But not all responded. Why? I want response from everyone. Okay, so most near about 56% responded B and 11% responded for answer D. But again, not 100% responded. Why? Okay, so the right answer for this question is, see, I have told you that in the protein synthesis, what are the important machineries, or we can say the tools required to charge the tRNA? It required amino acid first of all. It required ATP, that is adenosine triphosphate. Magnesium is required. I've not talked about the calcium, right? Potassium, sorry. So potassium is over here, so you can just cross this because potassium is not required. So magnesium, then there is a enzymes and tRNA. So definitely B is the right answer, and A and C adds ZTP is not required, so this is also can get eliminated. Now amino acid, ribosomes, initiation codon, releasing factor. Why releasing factor? Releasing factor require the last to release the two ribosomes, right? Larger and smaller subunits. So this is in case of elongations. It is not in the case of charged tRNA. So this can also be eliminated. So only B is the right answer. Now the next questions. In 225 amino acid sequence, if the codon for 27th amino acid is mutated to UAA, then what? You want me to generate the poll, or you want to respond in the chat box? Okay, how many of you like to generate the poll? You want me to generate the poll? What? Come on, respond it. You want me to generate the poll, or you want to give your answer in the chat box? If it's in the chat box, then you can give it in the chat box, no problem. Okay, so Anne said B, a polypeptide peptide with a 26 amino acid. Okay, why B? Surgeon. Okay, Anmoldeep responded A, polypeptide with a 2 to 4 amino acid. My God.
So first I got
Reza.
confused with 26 and 27.
Okay.
Then I realized first codon will be for start codon, so it cannot be taken into account. So it is 26 amino acids.
Okay, see here given as 2 to 5. So we are not interested in this complete long chain of 2 to 5. We are interested if the codon for 24th amino acid is mutant. That means of mutant with UAA. So UAA means what? This is what, a stop codon? Yes or no? Yes or no? What is UAA? So UAA the process of translation will be terminated because UAA is a stop codon. So therefore, the translation of 2 to 5 amino acid sequence will be terminated at 26 amino acid, right? It is 26 amino acid, because 27th is a amino acid which is stop codon. So if there is a stop codon, so it is going to be dissociate on that 26 position. So 26 before 26, 24 is 27, there is a 26, so total polypeptide with a 26 amino acid. So that's why B is the right answer. Okay, another question. Genetic code is universal. Genetic code is same for all organism. Now you have to tell me the answer. I think so both answer is, no, I'm not going to tell the answer, you just tell me the answer. It's a very simple question. Yes. Now I'm very clear with this two statement. Genetic code is universal, which is nearly universal we can say, because we have already studied, and genetic code is same for all organism. Universal, the mean is itself that it is applied to all type of organisms. So both statement can be called it as a true, right? So both assertions and regions are true, and the region is the correct explanation for of assertion. So yes, definitely A is the right answer. Yes or no? Then why not responding? Just Bhuwana responded. Okay, let's move ahead and try to understand the next question. I think so this is the last one.
Confused with nearly
triplet.
universal, sir.
Yes.
Nearly universal. The option was not there for A is false, so I got confused. Then you said you can take it as nearly universal.
Okay. Okay, fine, no problem. Now let's move with the next question. The triplet on coding strand of DNA is ATG. What would be the required anticodon on corresponding tRNA during the translation if there is a ATG? Yes. Okay, so give me the right explanation that why it is UAC. Who is going to give me the explanation for on this question why it is UAC? Because all are responded D. So tell me the answer. Oh, Prasant is also responded D.
Sir, in complementary base pairing, A will
Yeah.
always bind with T in case of DNA and U in case of RNA. Here, during translation, it is obviously RNA. So for A, it will be U. For T, it should be A. And for G, guana, it should be cytosine. So UAC.
Yes, it's absolutely right. So it is UAC. Very good. So Sarjana and responded nicely for this. They gave very nice explanation for both. As you can see, it is already written. Now this is a summary what we studied today. We studied about the genetic code, and then genetic code, the history behind the genetic code by George Gamow, then Hargobind Khurana, and then Marcel Nirenberg. Then we also studied about the salient features of genetic code, which include the triplet, then unambiguous and specific, degenerate, non-punctuations, then nearly universal, then followed by UAG act as initiator codon, also code for amino acid and different amino acid. We also studied about the translation of proteins. So these are the starting material for the translations. Then inside we also studied about the P site, A site, E site, initiation codon, stop codon, initiation factors, elongations, and releasing factors. All these are important that you have to remember. Okay, so these are just a summary that what we studied in the translation protein synthesis process and also in the genetic code. Now it's time to move on your dashboard and try to solve the class assessment. So I request all of you to please move on your dashboard, and there is class assessment with the name of genetic code and translation N4, and all of you please attend this question because these questions will reflect on your result, because you know that there is a PTM, parent teacher meeting, on every month. So it is going to be noted in that whether you are attending, not attending, responding, not responding, everything is going to be monitored. So please be what alert the time of attending the live sessions and also for class assessment. So once the session is visible, please start attending the I mean to say the class assessment. Just check your dashboard and tell me whether that class is visible to you or not. Sarjana, okay, visible sir. Okay, if it is visible, then start attending. Now today you have ample amount of time, 20 minutes you have in your hand to solve only hardly 10 questions, right? So those who got it, please start attending that class assessment and do it fast. So today's session is based on the what we can say theoretics. It's very theoretical, so you have to remember that because this is important and you have to be remember. Yes, do it fast. Okay, so Anmol Deep and Sarjana. Okay, done. So tell me any difficulties in that questions, or if you feel that this question is out of box and something like that, so you can. Yes, anyone? And no, I think so all question up to the mark and up to the syllabus for today's syllabus which we taught, which we studied. Sir, can you okay, Sajana again said, sir, can you explain the 7th questions? Okay, so now I have to go on and find the 7th question. So the 7th question, class assessment with the question. Yes. So 7th question, can you read the 7th question, Sajana? Because question number can be vary, na? Like.
Sir, yes, sir. So for me, the seventh question is vowel position means base pairing, second altered base and codon. third altered base on codon and none of the above.
Okay. Okay, got it. So okay. Let me first of all tell you, vowel hypothesis is what exactly? Okay. So basically, we all studied about the cloverleaf model. Yes and no? Yes. So vowel hypothesis is what actually? It is also a similar kind, like as we know that in the vowel hypothesis, it is a hypothesis. Okay, vowel basically name of the scientist who hypothesized the statement that at the base at fidus end of anticodon is not confined to the other two bases, means either initially it is UA, C or Cu. That means of other two bases allowing it to form a hydrogen bond, and any one of the several bases located at the 3' end of the codon is a vowel hypothesis, which is hypothesized by the vowel. Okay, that means of in your questions what is given in your questions, it is given like that. The vowel position means what? So always keep in mind vowel position is that means of it's a third nucleotide in the codon. So that means of in the nucleotide codon, the third position, that vowel position of codon refer to the third nucleotide in the codon. So according to this vowel hypothesis, only the first two base of codon have a precise pairing. Now what is mean of first two codons? So you can see here, suppose this is U, then there is C, and then there is U. So initial two, okay, suppose the initial two is what? This initial two referred as these two in initial two bases of codon have a precise pairing with the base of anticodon of tRNA, while the pairing between the third base of codon and anticodon may be vowel. That means of this codon may be vowel. This is third position and this is the first and second. So first second position can be code anticodon accordingly, but this one is can be a vowel position. So this is a vowel positions, and that is according to the question that which position means a vowel position. So it is a third alter base on the codon, which is referred as a vowel positions. Clear? So in any of this code, the third one is a vowel position, always referred.
Yes, sir. Got it, sir. Yes, sir.
Clear, na? Okay. Any other question? No? Okay. Then, well, if there is no question, then I'm going to wind up this session. Thank you very much. And then Sarjana, then Prasanti, then Anmol Deep for your joining now. I'm going to wind up this session. In the next session, we will try to learn the another concept, that is gene regulation, and something that we will see in the next upcoming session on Saturday, I think so. Yes. So for the day, bye. Take care. We'll meet on next time, and don't forget to complete your homework also, because homework is also assigned to you. Yes. Take care.